So for example, you can have neurons that are photoreceptors that detect light of a given wavelength. You can have neurons which are specialized in processing information for motion detection. You can have other neurons which are involved in processing color vision or polarized light vision. And all those neurons are in the brain of the fruit flies, and each one of them seems to have a special, or a group of them have special features that allows them to do that. So how can we use our big knowledge of what the brain, how the brain of the fruit fly is organized, and go one step further and analyze what's happening inside of each one of those neurons?
Since we've been here, we have done a few things with generating ways of activating neural circuits within the brain. So what we do is we take native neurotransmitters such as dopamine or serotonin, and we make modifications to them so that they're no longer active. And then when one shines light on those molecules within a brain, in an animal, or in a tissue slice, or in cell culture, we can then activate that neurotransmitter. So it can turn on the circuits that neurotransmitter turns on.
We are interested in vision, and we use actually a model system, which is the fruit flies, what you see around us, to study what's going on there. The reason for that is because, we can manipulate this organism and understand what happens. And I always tell people that, if you wonder why we should study fruit flies for vision, just try to catch a flies, and you'll see that most likely they have a better vision than we do. And in fact, we share a lot of mechanisms of vision with the fruit flies, like with many animals. So by understanding what happened in the fruit flies, we hope to be able to understand what happened in higher organisms, and including in humans.
We use fluorescence as a tool. A lot of our stuff would be more related to activating things with light. And so yes, Abu Dhabi is enormously sunny. Why don't we just take some of our reactions outside and put them in the sunlight and monitor them and see how we can get them to go in sunlight rather than the controlled environment with an LED lamp or a laser.
We label the peptides or the proteins that we use as delivery tools, or the drugs themselves, with a dye, essentially a fluorescent molecule. And using a fluorescence microscope, we then track it as it interacts with the cell, and then as it gets into the cell, hopefully, and where it ends up in the cell.
A lot of our work is in the microscopy lab using the confocal microscopes, which are used for looking at fluorescence activity in cells. And corals naturally generate the fluorescence that a lot of people use, for example, as cell markers in other areas of biology. So they're actually taking a green fluorescent protein that actually originally came from a close relative of corals, the jellyfish. We naturally have these occurring in our tissues. And so we use that sort of equipment to look at fluorescent proteins, as well as other equipment for looking at chromoproteins, colored proteins that are in the coral tissues themselves.
And I still remember the day that I was looking at one of my gel results through basically looking at what is an X-ray film and looking at it against the light, and in came my friend, who's this artist, and he just looked at me and I looked at him, and he said, "I know you think that's data, but I know better," because he was really looking at the patterns, and then he took it and made an art piece out of it after I analyzed it. That actually was published recently in the Journal of Material Chemistry C, and we were selected to give the cover. And as you can see here, we have this molecule here, where we can switch its properties between transparent to opaque, and you can by switching on and off, you can block the sunlight, and you can save energy. And actually, I'm not sure if I have it. We built a little device, like what we call a smart window, where we were able to block, or we can switch it on and off like cycles and cycles without harming the property of this window. And you can see we go from transparent to purple. By blocking the sunlight, you don't have to use your AC as much as you use it on everyday life.
This is X and Y are positioned on the sky, and the color is the number of photons. Black is more photons, white is fewer. You notice a lot of them are just nice simple circles. So those are point sources. This one is extended, and since it's not within the plane of our galaxy, it's almost certainly a galaxy cluster. And the fact, normally a galaxy cluster is brightest in the middle and has sort of a smooth drop off in brightness as you go further out. And the fact that this one isn't circular or elliptical at all, and the brightest point is not at the center of the object, and there's sort of two brighter spots suggests that there's actually two clusters which are overlapping and possibly integrating.
Do I need to explain what black holes are? Or? Black holes are objects where the density is so high that even light cannot escape from them. So for example, from Earth, rockets, if they go fast enough, can escape the gravitational field of the Earth. And for different size objects, they have different speeds at which you can escape its gravitational pull. And the fastest thing anything can go in the universe is light in a vacuum. Nothing is allowed to go faster than that. But these objects have such strong gravity that even light cannot escape from their gravitational pull. So whatever falls into the black hole just stays there.